Welcome, friends. We're here today with Aubrey de Grey, Natalie Coles, and Jose Cordero, who very kindly accepted to talk to us today, and David Garcia Monach and myself to represent Alianza Futurista at the moment. We don't have anybody else who could come today, but we'll just go ahead with our conversation with these wonderful guests. Just to mention for the public, I've known Natalie for like 15 years. I've known you for like 20 years. I think Transvision 2006 was the first time we met, which you brought me there too. And so we've known each other for a very long time. We follow the progress of everything that's been going on in everybody's lives. But we've never gotten the opportunity to talk in this kind of setting. Jose Luis, what's happening with your Stop Envejecimiento?
Well, we are trying to stop aging in Madrid, where we are based, Spain and the world. And there are some people who are experts on that, like Aubrey de Grey. That is why we are so pleased that he's here. And not Aubrey to stop aging, but also Natalie, because she's an expert on supercentenarians. Until about a month ago, the oldest living person in the world was a Spanish woman who lived 117 years and 168 days. Longest living person certified in Spain by the Guinness World Record, and the 8th person in the world. All of you know Jean Calment, who lived 122 years and a half, and then other people, and then Spain. So we are very happy to have this in Spain, and Alianza Futurista is doing great things to promote these ideas that we can live not for 117 years, for 117,000 years, 117 million years.
Yeah, the goal is to live indefinitely, and I'm not sure most people realize that's what we're aiming for, because sometimes people in the field are a little shy about opening up this kind of conversation. Like, we really want to end aging. It's not 120 years, 140 years. No, we want to get rid of aging.
Yes.
And we're very determined. We know that there is a lot of research efforts going on all over the world. I suppose especially in the US. I suppose it's based very much, I would guess. I'm not sure if you can educate me, but I know a lot of the effort is in the US. And one of the questions that I've been thinking lately, just out of curiosity, if you know, just out of hand, is there any or a lot of public investment in this problem in the US? Or
so when you say public, you mean from the government?
Yeah.
It's getting better, but there's certainly not nearly enough. At the moment, the public investment in medical research in general is almost entirely funneled through an organization called the National Institutes of Health, which has a component called the National Institute on Aging. But if you look into the details, it's not really true. Hardly any, or maybe 15% of the money that the National Institute on Aging spends goes to medical research on the biology of aging. And out of that, only a very small minority goes on to the efforts to actually do something about aging. So it's really, really bad. But one piece of very good news is the creation over the past few years of a new thing called ARPA-H, which is an agency of the US government designed along the same lines as the much better known DARPA. Essentially, it's focused on doing really visionary, longer term stuff. And the H stands for health. So this is very much progress. And sure enough, as this agency is coming into existence and getting shit done, it is beginning to lean towards doing something serious about aging. Very recently, it hired a program director. They have only a couple of dozen of these. A program director who's one of the most visionary academics in the field. This is the kind of thing that gives me hope, but it's still got a long way to go.
So we would say that most of the investment comes in the private sector?
Well, yes, but that has to be qualified as well. The private sector investment in longevity was more or less non-existent until about 6 or 8 years ago. And then it started to pick up in a big way, and we were very happy about that. But it was at the level of very early stage stuff. Angel investors, seed investors writing 6-digit, 7-digit checks. Nothing huge. no interest from institutional investors or big pharma or anything like that. Then after a couple of years, there were occasional, gleams of light. Some institutional investors got involved in one particular company called BioAge, which has done very well ever since and is actually in the process of going public right now. Public in the other sense, of course, the word being listed on the stock market. That has continued, and certainly we've since the past couple of years seen very large amounts of money coming in from wealthy individuals, of which the biggest is Jeff Bezos putting $3 billion into a company named Altos Labs. But there are plenty of other 9-digit ones. So this is really good news. However, the qualification is that it's very, very unevenly distributed. The things that are getting the money are things that people believe they can make money out of fairly soon, the low-hanging fruit, if you like. But aging, being a very complicated, multifaceted problem, demands a divide and conquer solution.
Focused effort.
No, a non-focused effort, a multi-focused effort, that's the point. A divide and conquer solution. And that means that there are going to be areas which are more difficult, less quickly monetizable, less attractive to investors. And the result, of course, is that they don't get so much money. So right now, as I say, very unevenly distributed. There are vital areas of this research that are still really, really, really struggling for money from either the private sector or the public sector.
By focused, I mean like a Project Manhattan, Project Apollo.
So at the moment, we would like to have an Apollo project for aging, but you don't really need it. An Apollo project, it kind of implies that, yes, you've got lots of components, but everyone's working together under common leadership and all that kind of thing. That will be fine, but it's not actually required. We are sufficiently cohesive as a community that we can do it without the overarching leadership, just so long as the money flows to all the places it's needed rather than only to some of them.
If you want to...
Yeah, actually, I would like to ask you about the RMR project.
Okay.
And I have here your last Twitter post, I think it was from August, about the update. And I was wondering if you could expand on some of your conclusions, like why was this analytic branch bust?
Okay, well, so I think I owe it to the audience to give at least a few sentences of background in order to give them any idea what the hell you just said, rather than just launch into an answer.
Yeah, sure.
So the project that you're describing is the flagship research project of the foundation that I currently lead, LEV Foundation, Longevity Escape Velocity Foundation. And the goal of this project, and projects like it that we want to do going forward, is to combine interventions in mice that already work as good rejuvenators to some extent, and to see what happens when we apply all of them to the same mice at the same time. So at the moment, we are taking 4 such interventions. Now, the key thing is they've got to be rejuvenating, or at least they've got to work starting late. In other words, if you don't do anything to your mice until they're already in middle age, then, so they've got like 1 year to live.
After 1 year of life?
1 year of life to live, that's right. So they don't typically
1
mice...
year to live.
That's right.
Let's get out on you.
That's right. So typically these are mice that live an average of about 2.5 years normally. So we start our studies when the mice are already 1.5 years old. And so we want to try and extend that. And of course, the extension will be extension of healthy life, not of the end of life when the mice are unhappy. That kind of goes without saying, though a lot of people get it wrong. So that's what we're doing. And historically, the best that anyone has achieved, starting at 18 months of age, is about 3 or 4 months of additional life. We want eventually to reach a whole year of additional life. We want to get out to an average lifespan of 3.5 years, and that would equate typically to a maximum lifespan around 4.25. And we certainly won't do that with this study that's happening now, but we'll probably beat the record for whatever's been achieved before. So we're pretty happy about that. We feel we've definitely justified the motivation for the study, the idea that this is really the most important, the best value for money study that anyone can do right now in the biology of aging. And that's a big statement because these studies are very expensive. They take 1,000 mice, So anyway, to come back to your questions, we've learnt quite a lot from this study. As I say, we have learnt that the study makes sense. In other words, that we are seeing, additive contributions from different interventions. We've had one or two disappointments. One disappointment is that our senolytic doesn't seem to have worked. That is the drug that we gave these mice to try to eliminate the cells that are accumulating in their body that have this characteristic called senescence, where they are toxic, they secrete nasty things. And we feel, we don't know exactly why it didn't work, but our best hypothesis is that we were essentially misled twice by the literature. We had to believe in the literature up to a point, right? And essentially it was a problem of solubility. You have to dissolve your drug.
This moiety of galactose plus navitoclax, right?
Right, so the senolytic we used was something called navitoclax, about which there is good evidence in the literature that it works reliably, and that's what we really wanted. A lot of the other senolytics, there's a lot of shortcomings of reproducibility. And the thing about navitoclax is that it was known that there was one particular cell type that we don't want to kill, that it did kill, and that was platelets. Several years ago, a Spanish group figured out a really clever way to prevent that from happening, which was to conjugate the navitoclax to a molecule of galactose. Galactose is a sugar, and it turns out that senescent cells almost always have really high levels of an enzyme that breaks down galactose. So, in other words, this thing that we make is a prodrug. It's inactive against senescent cells in its original form, but when it gets into senescent cells that are expressing this high level of galactose, which platelets do not do, then it gets activated by the removal of the galactose, and lo and behold, the senescent cell gets killed. That's fine in theory, but in our hands, it didn't work. We couldn't get it to dissolve well in any of the solvents that had been talked about.
Because it was taken orally by the...
But originally we wanted to inject it in order to get it to cells, and we couldn't dissolve it. So eventually we found a solvent which did work, which did dissolve it, but the mice really didn't like that solvent. They got sick just from the solvent, right? So we eventually had to retreat, as you say, to delivering this thing orally, and that's a problem, because it's a fairly big molecule, it's going to get broken down in the gut, it's not much, very little of it is going to get into the circulation. So that's our best hypothesis right now for why it didn't work. We just got unlucky with the incomplete information provided in the literature. Too bad. These things happen in experiments all the time, and we feel quite lucky that it only happened once in our experiment. Other interventions worked fine.
And actually, I was also curious why, because I would say that not all of these approaches are purely sense-based.
Well,
so I guess you made your approach wider.
So our take was, we've got a lot of choice. At the beginning, when we were designing this experiment 2 years ago, we said, well, okay, what are we going to include? And we had so much choice. So we decided that, well, one really big criterion is going to be direct evidence from the literature that this really works when you start late, and it works in mean and maximum lifespan. And another big criterion would be that it would be a sense intervention, a damage repair intervention. Eventually we decided that we were going to compromise on that second thing in relation to one intervention, namely rapamycin. We were going to put rapamycin in anyway, even though it slows aging down rather than repairing damage. And the reason we said that was because that was the intervention that had by far the best evidence for actually working, right? All the other ones we were including would be ones that, there were a couple of papers that looked like they were saying, that we're getting a good increase in lifespan. And rapamycin, there were like a dozen. So we couldn't leave it out. That was our rationale. Our next experiment, what we're expecting we'll do, we haven't completely decided, but our current thinking is that we'll actually just give rapamycin to everybody. Because, that's right. Because, it's established, it works, and we want a baseline that is as good as possible so as to give a better, if you like, signal-to-noise ratio for the interventions, for the damage repair interventions that we're actually testing.
Actually, I'm also curious that you said that for, I don't know if it was female or males, it's like these damage repair interventions, they diminished the effect of rapamycin.
Okay, so yes, so this is something I don't want to be too definitive about yet, because really it has to wait until the experiment is completely over, and we've still got 50 mice left out of the original 1,000. But yeah, so if you look at the curves for females, everything looks really clean. It's pretty clear what's going on. It's pretty additive. The only bad news with females is that most of the signal seems to come from rapamycin. In other words, the 3 damage repair interventions are not really adding very much. And what that tells me, and I say this is tentative, right? What that tells me is that the things that kill mice are not all being hit by our damage repair interventions. There are some things that are just being missed. Now, we know that our damage repair interventions are hitting different parts of aging, but only that their direct targets are just a subset of aging. But we are relying that there will also be knock-on indirect effects beyond the things that we are directly hitting. It looks as though in females, the combination of the direct plus the indirect ones were still left things out that just were killing the mice on schedule, if you like, or at least not quite on schedule, but on schedule once you'd put the rapamycin in. All right, so that's the story with females as we currently have it. Story with males is much more complicated. First of all, we have a couple of treatment groups that were getting 3 out of the 4 interventions, and yet they were doing considerably worse than the control group that wasn't getting any of the interventions. Crazy, right? I mean, and we still don't understand that. Especially, we really don't understand it now because that was only true for the first 9 months of the study. In the second 9 months that have happened recently, those groups have caught up and they're now doing better than controls. So we don't know what's going on. And, this is the way things, you expect to be given surprises in experiments, right? That's why you do them. But coming back to the interpretation of females, when we look at the effect of things over and above rapamycin, unlike females, we are seeing a very significant additional effect. So if we look at where the group that's only getting rapamycin is, in females it's doing almost as well as the group that's getting everything. But in males it's doing hardly any better than the group that's getting nothing. And the group that's getting everything is doing a lot better than that.
And I think you already had into account that so far males for mice were doing worse on rapamycin than females. So you took a higher dose of rapamycin for both female and males? Or?
We used the same dose for both sexes.
It was higher than the usual dose for males.
Actually, no. So in the literature, there are a lot of studies at 14 parts per million, and a lot of studies at 42 parts per million, 3 times as much, and also a few studies at 3 times that, at 146 parts per million. We're using the middle dose, 42. That's been used a lot, and it's been shown to work quite well. So we believed that was the right dose, and we still do believe that.
But have you checked the rapamycin blood levels in both females?
We have not checked that yet. We're storing plasma from these mice, so we have the opportunity to check that. But at this point, it's definitely working. We're getting definite results in both males and females.
But I think the open question for rapamycin is it that females absorb more rapamycin, or is it something metabolically
Sure.
that for females it works better than for males?
Sure, that's a big question. And of course, it's a question that can be asked outside of the kind of studies that we're doing in the context of other interventions being overlaid on top. So these are questions that we don't really feel are important for us to answer because they're being asked by other people who are focused only on rapamycin.
Let's ask Natalie for a minute to describe for us in a few words, so people who didn't see your talk yesterday, etc., who are not going to take the time at home, average people, what do you do with super centenarians? Could you explain in a few words?
Sure. So, well, currently, I think it depends on who I'm working with at the time. But basically, for the most part, generally what we do is I do biological sampling on the world's oldest people. So people who live to be 110 or older. And if they consent, we do blood samples, and we also do blood samples of their offspring, so their children. And if they consent and the right team is in place, we try to do autopsy. And so I've worked with multiple groups on this, and depending on who's funding and what they're looking for, different studies have taken place. So in the past, I worked with Steve Horvath at UCLA, sorry. And during that particular study, he did find, and we published, that supercentenarians, they found during autopsy, one, that they tend to die of a protein buildup called TTR amyloidosis, which was surprising. The other thing that we found was that the epigenetic clock within the cerebellum of the supercentenarians ages slower than that of the regular population. And then we did another study with Stanford, which was separate, which way they tried to do sequencing and find out if they had anything in common. At the time, this was like over 10 years ago, so the technology wasn't where we are now, and therefore we didn't find anything that was enough for us to publish. But the problem is that when we do this cohort, it's such a small cohort that they're compared to the normal population. And so one of the goals that I'm working on right now is that if we can get enough supercentenarians, they will be able to make them their own cohort and measure the regular population against the supercentenarians. So one of the things we found with the Stanford study was we did find areas that we found interesting, but not significant enough to publish. So the public doesn't know about this, but two areas that we thought might need a little bit more research were heat shock genes and killer T cells.
What is heat shock gene?
So it's hard for me to explain, but Aubrey probably explain it a bit better with the heat shock genes, but they're involved with a lot of sort of immune response and inflammation.
Like so much in biology, heat shock genes are named in a misleading way because names are often arise for historical reasons. These are called heat shock genes simply because they were first discovered in fruit flies decades and decades ago as a result of the way that fruit flies respond to high temperature. But in fact, they have much more general functionality. They are what's often called chaperone proteins. So they help other proteins to maintain their correct shape in the context of stresses of all kinds, including heat.
Yeah. And so the last two things that I'm going to say in regards to the research that we're doing now is one thing that we didn't anticipate, but we've been noticing is that when COVID hit, I thought all this whole population was going to pass away and therefore the research wouldn't continue. But now that we have resumed it, we have found out that the supercentenarians that are alive that I've sampled, not only did they get COVID once or twice, but they survived it and they didn't get hospitalized. And the hypothesis, we haven't confirmed it yet, more research needs to be done, is that they were actually children during the Spanish flu or potentially in the mother's womb during the Spanish flu, and therefore they had some sort of exposure or antibodies or immune memory, and therefore they were not affected as deeply as the other people.
Yeah, it's going to be really interesting to test that hypothesis over the coming decade,
Right.
because the people who are the very oldest in the world now actually were already like 10 years old at the time of the Spanish flu, whereas the real question about things like prenatal exposure will only be possible to ask when the people who were actually in the womb in 1918 get
Right.
to be. Yeah.
And another way to sort of figure out whether or not that might be accurate is to start paying more attention to supercentenarians in Asia, because they were not as exposed to the Spanish flu.
Right.
So to see, do they have a higher rate of supercentenarians that have survived COVID? And if not, then that would be indicative of the fact that maybe that hypothesis is true, that's why they did so well. And then the last thing is currently, so the teams that I'm working with now, where one of the teams is not really getting any data, mainly because of funding. So that's another thing. But what we're doing is trying to create a biobank of the world's oldest people so that we can immortalize the cell lines. And people ask, why would you want to do that? Well, the thing is that if these cells contain anything including that is relative to longevity, and we don't know exactly what that is, we're now at a place where we can actually start creating organs and things like that and if we use these cells, theoretically, they would contain the properties, and therefore, not only, even if it's not a human, if it's just an organ, they might contain properties that have them be long lasting, right? And lastly, there's a team that currently I'm working with samples with one at the Buck who's doing anti-gravity with astronauts and seeing if supercentenarians have some relative ability to withstand that. And then another one, well, what they want to do is get supercentenarian cells, cells of the offspring, reprogram those cells into different tissue types, and do that with a regular public person and see, is there particular organs or parts of the body that go slower compared to other. And then lastly, we know that there were people that, I think this was published through Ajax, but maybe you can correct me if I'm wrong, is that they were able to get one of supercentenarian cells and reprogram it back to a pluripotent state. So basically get one of the oldest cells possible and bring it back to zero. And so that's a big deal for a lot of people and for us. And if we know we can do that, then that's a step towards longevity and being able.
Yamanaka.
Yeah, Yamanaka factors.
Very old.
Yeah, and starting it back.
It's so the original Yamanaka factors are pretty good at rejuvenating cells that are already differentiated, but cells that are particularly old, including cells that are senescent, that occur, as I mentioned earlier, in younger people, these are harder to do. And so people found additional factors that you could put in, ways that you could optimize the de-differentiation process.
Have we been able to stop this going back to? Because I think some of the problem is that you reverse all the way to the beginning, but you want to.
So, okay, there are two answers to that. One question is it possible to take a differentiated cell and take it back so that it's regenerative but not truly pluripotent? The pluripotent cell is one that can do absolutely anything except make a placenta. You have to go even further to get to that. But pluripotent cells can do anything, and so you don't want to put them in the body, because when you do so, they don't know what they ought to do. They have signals from their environment, but their environment is so different from what they are evolved to respond to, namely the early embryo, that they don't know what to do. And they tend to create an unusual kind of tumor called a teratoma, in which you have a lot of different tissues all in the same place at the same time. You have teeth growing in heart tissue, things like that. Literally, I'm not even exaggerating that. People often think I am, but I'm not. So that's the problem with pluripotent cells. So what do you do about that? There's two options. One is that you stop the de-differentiation process, the winding the clock backwards, when it's got to less far than that, when the cells are regenerative but not truly pluripotent. And that turns out to be hard, but there have been clues recently. David Sinclair, among others, found that if you leave out one of the 4 original Yamanaka factors, a gene called c-Myc, and you only use the other 3, then it looks like they kind of hang up around the right time, which is quite nice. Now, that's not good enough, though, because you still want to be able to tell the cells which way to go. And this is the topic of some fantastic technology that was developed by a real godfather of the field, Mike West, which is now called PureStem. And essentially it's a way of creating adult stem cells that are not so...
PureStem?
Yes, PureStem. P-U-R-E-S-T-E-M. It used to have another name that was harder to pronounce. So this is a way of creating really high purity cells of whatever type you like, including any kind of adult stem cell. It's a tragedy that this technology has not moved forward as fast as it could. Very recently, there are other approaches that appear to be better than what we could do before in terms of doing the same kind of thing, but I've not heard of any report of people getting anything like the same purity. And purity is quite important. You do not want to take a bunch of stem cells that are mostly the right thing, but some of them are the wrong thing, and just inject them into the body, because you never know what's going to happen.
Jose Luis, feel free to ask any questions to our guests.
Well, we are very happy to have Aubrey and Natalie here in Madrid, in Spain. Actually, we were going to go to meet again the oldest living person, Maria Brañas Morera, who lived in Catalonia. Unfortunately, after the age of 117 years and 168 days, she died on August 19. So we did not have that opportunity, which was very important to see how supercentenarians also evolve and to sequence them.
But she was already sequenced.
Yeah, the first time. No, but the idea is to do it again and again.
Oh, right.
I mean, of course, the DNA sequence stays the same, but the epigenome changes.
Well, first and foremost, let me just clarify, because I think this is a huge misconception as to what's happening now. And I feel if I don't clarify it, then it seems like the research isn't being done properly. The truth is that right now what we're doing is banking the cells. So there's this misconception that people are doing a lot of studies and no one's doing any studies. There's zero data. They are using the cells and immortalizing them and putting them in a bank. What we can do with that is basically what I just told you with the studies of being able to reprogram those cells and then having a race and seeing what is it. But as far as a lot of people ask me, well, what did you get from there? We're not doing any stuff right now, right? And
So you're collecting so that
yeah, and
either others or yourself can then go into
we're hoping, but the thing is, once you do an immortalization, you can't really test these biomarkers because they're all back reset. Do you see what I'm saying? So we can't. When you immortalize a cell line, you're bringing it back to this, like, this basic state where they're new cells. They're not. So you can't get those cells and start doing biomarkers on them because they're back to a younger state. They're not at the state in which I drew them.
This is very important, and I
Right.
totally agree with you. This is fundamental to understand this and what we can do. But talking about Madrid,
Right.
there is all this discussion about blue zones.
Yeah.
And I think this is important because we discover things about nutrition, lifestyle, and how people live. But, Madrid has the longest life expectancy of any European capital. It is 84 and a half years. And Spain as a whole has the longest life expectancy of any country in the European Union. So when we look at the blue zones, and in Europe there are two, the island of Ircadia in Greece or the island of Sardinia in Italy, Madrid has a longer life expectancy than Icaria or than Sardinia. So my point, if these places are blue zones, then Madrid is a super blue zone.
Yeah.
And also because in the bigger Madrid, we are 8 million people. 8 million people. If you look at the population of Icaria, it's a statistical mistake. Or even Sardinia. It's not a really big
So I need to correct you a little bit on this. So no, it's definitely not a statistical mistake.
It's a joke.
The difference between
the joke here because
the difference between the actual blue zones versus somewhere like Madrid is that the actual blue zones have a substantial difference in average lifespan between their population and the population immediately surrounding them. So the fact that they are small is the point. Whereas you, as you just said, have the exact opposite situation where, yes, Madrid is doing really, really well, but so is the entire country, right? So the difference between Madrid versus the rest of the country is actually not very much. That's why it's not a blue zone.
And you have to take into account that there's a lot of smoking in Spain too. So if you actually take into account that, 40% of the people smoke cigarettes, then that it would probably be higher if there weren't so many smokers, that average.
Yeah.
That includes everyone who smokes as well, I suppose.
I think
the averages.
this is an important point for me, though, because I feel like there was just recently a paper about the blue zones, debunking the blue zones. And a lot of people, in my opinion, this is a personal opinion, were throwing the baby out with the bathwater. And what I mean by that is people were saying, well, these blue zones are fake because it doesn't really matter where you live. And I think that it's the way you interpret it. If you say, oh, well, I'm going to live in this location and therefore I should live this much time. That's wrong. What they're trying to it's a blue zone because if you live amongst this culture of people and take in these sort of habits or lifestyles, they tend to promote longevity and you live a long time. And therefore, so if it, depending on how you look at it, Madrid in itself could be a blue zone. It may be the biggest blue zone. And the reason I say that is because, well, if you look at it from a perspective of the culture and the things that it encompasses, smoking, of course, is bad. But we do notice, and maybe it's a genetic thing, but what we do notice is naps are good. Coming out at night and hanging out with your friends, that's good. One-on-one, not being lonely, that's good. Having olive oil, that's good. Having immense respect for the elderly and not putting them in nursing home, that's good. So these are things that it's not basically, well, I'm gonna move to this location and because I'm on this little piece of land, I'm gonna live this long. No, but if you incorporate the sort of traditions of this culture that promote longevity, then that's what a blue zone is, in my opinion. That's my own opinion.
But let me go beyond blue zones,
Okay.
because I love the concept and I like the idea of centenarians and super centenarians.
Right.
But our goal should be biological rejuvenation.
Absolutely.
I
yeah.
don't want to be a super centenarian the way a super centenarian is. I want to be a super centenarian at a biological age of 2025.
I guess they study, I mean, just like you study very long living animals, it's all data that's useful for the research.
Sure, and this is fantastic because even among mammals, we have small shrews that live 2 years, and we have bowhead whales that live 200 years. And we share over 90% of the genome between shrews and mice and monkeys and elephants and whales. So why do we have whales that live 200 years? I want to live 200 years at least.
Yeah, so, I mean, one thing we have to always bear in mind is that these are two overlapping but somewhat distinct fields of research. The research into why aging goes at different speeds is one thing. The research into reversing aging is a completely different thing. And yes, there is some overlap because we need to understand what we are reversing. But this takes me back to the very beginning of my work in building this community 20 odd years ago when I started running conferences. I brought together people who would call themselves gerontologists, who would say that they work on aging, with people who did not call themselves gerontologists. They would say that they worked on stem cell research or tissue engineering or bioremediation or something. And literally half a dozen of the speakers that I invited to my first or second meeting would write back and say, well, thank you very much, but I don't work on aging. Why the hell are you inviting me? And I would have to actually explain all this to them in order to get them to come. But of course, this was absolutely the formula that was most important, except perhaps the free bar, that in making my conferences so successful. It was bringing people together who did not know each other's work previously, and who formed new collaborations, new alliances, new friendships. Of course, this applied also outside of the speakers. It applied to all the delegates. So, I feel that was a very good move, but it was really driven by the nature of the problem, that it was being studied as a phenomenon rather than as an engineering problem. And as such, people were trying to figure out things like, okay, why do different species age at different rates? And I came at it as an engineer and said, well, okay, we need to apply regenerative medicine, broadly defined, to the problem. And that was a hard sell. It took me several years.
Predictions. What are your predictions? Kurzweil is relatively optimistic. 5 years, 15 years, we're going to reach longevity.
Yeah, 20 years. No, first, in 2030, we reach longevity escape velocity.
5 years.
And in 2045, we rejuvenate people.
Yeah, which honestly, I need to have a word with Ray about that, because the fact is, it is embarrassing to have two different numbers there, given that the way we're going to get to longevity escape velocity is by rejuvenation.
Yeah.
So I need to explain. I need to get him to actually, sort that out.
No, but I see a difference. I mean, by 2030, we will be living longer and longer, but it's still aging.
No, the whole point. No, the whole point. I don't know what we're going to be at, where we're going to be at by 2030, but I do know that when we reach longevity escape velocity, by definition, we will not be aging. We will not have an increasing biological age. And the way we will do that is by combating biological aging by rejuvenation that is going faster than the aging.
So you're cycling, you're rejuvenating. letting sometime that rejuvenate again. It's going to be cyclical, like Sinclair.
Well, that's right. I mean, of course, the details are unclear. There may be some rejuvenation treatments that we will apply every week. There may be some treatments that we will apply only every 10 years. It just depends on the specifics. But the point is that our overall biological age, which we may as well define as the total amount of damage that our body carries, will not be going up. So in that sense, we will not be aging. And that is the definition of reaching longevity escape
It's
velocity.
probably not a good idea to make predictions. Just throw money at it, throw work at it.
Time frame predictions, yes, that's correct. I mean, it's a good idea. It's a very bad idea not to make any time frame predictions because people will just carry on thinking that it's just never going to happen ever. But it's also a bad idea to give time frame predictions that have some connotation of certainty when we're talking about pioneering technology. Clearly, it has to be probabilistic.
I do like predictions, and when I met the British science fiction author Sir Arthur C. Clarke, he said that these predictions are important, both positive and negative, because he said they become self-fulfilling prophecies.
Right.
Yeah.
So we do have to think about the future, hopefully in a positive way, and also into reaching longevity escape velocity so that they happen.
And this is well known, and even in terms of the speed. So the best example is Moore's law. Yes. If you talk to anyone in the computer industry about Moore's law, they will agree that the reason Moore's law has been so faithfully followed, that the doubling time has been pretty much constant for so long, is because it has become a goal of the industry. Right. The industry has set itself the goal of keeping up with that. It could have set itself a higher goal, and it would probably have reached it, but it didn't because Moore's law became a mantra.
I think also too, I like predictions for the reason that I think that there's a big part of the community or population that sort of is not really invested in longevity, but they follow it. They don't actually contribute. And the thing with that is it's good to have a prediction so that they believe that we are working. We do believe that. But at the same time, if we don't hit it, I think what's important is that it makes people sort of stop, pause, and remodel the problem. And I think that so many people that are not involved in the science are just so focused on the goal that they don't pay attention of how are we modeling the problem. And in order for you to go back and sort of remodel how you view of it and how you tackle the intricacies of it requires people sort of going back and reflecting upon that. And I think that's important.
So to what extent do you think that longevity escape velocity has increased since you coined the term like 20 years ago?
How do you mean increased? I mean, that's not the right word because clearly it's defined as the minimum rate at which we need to improve the comprehensiveness of the therapies. So I don't understand your question.
Yeah, I mean, if you compare
I think the question
now
like,
with 20 years ago,
are we closer to longevity?
Okay. If you're talking about how soon we will reach the longevity escape velocity, the point which someone named Paul Hynek called the Methuselarity. Mm-hmm. Yeah. So the situation is very good right now. So I now say it's going to. I think we have a 50-50 chance of getting there within 12 to 15 years. So that means the second half of the 2030s. Right. Now, if we go back 10 years ago, I was already saying the second half of the 2030s, right? Things have slipped maybe by a year, hardly at all in the past 10 years in terms of my predictions. If you look at the previous 10 years, however, it's an absolute clusterfuck. It's terrible. The predictions were slipping and slipping and slipping.
So you think the increase is still linear, or one thing you will get exponential, or?
That's again the wrong question. My point was that it was slipping in those first 10 years between about 2004 and about 2014 because of lack of money, not really because I was wrong about any of the science. And it hasn't. And the reason it hasn't slipped at all, to speak of, in the subsequent 10 years, is because we've had the money by and large. The money is very unevenly distributed, as I was saying earlier, but it's a lot better than it was. So, it's still a matter of a date at which we can achieve things. It's not a matter of longevity escape velocity increasing or decreasing. It's a case of will we get to the Methuselarity. Of course, once we get to the Methuselarity, once we have reached longevity escape velocity, we then have a second question, which is, what is the chance that we might fall back? We might suddenly have a really bad period of time and make little progress so that we end up aging again. And then we can say our chances of that are vanishingly small. And the large part of the reason for that is something that where your question kind of does make sense, namely, the further we get in terms of bringing aging under comprehensive medical control, the slower longevity escape velocity becomes, because we have less to fix. So the stuff that we still can't fix takes longer to catch up with us.
Yeah, I guess the right question would be, I mean, we are not yet at longevity escape velocity, but how fast are we getting to it?
Well, so yeah, I can only answer that question in terms of how soon I think we will get to it, right? And I've just given you that answer.
Actually, I have one question about it. I hope I can express it. But right now, I'm closest to the epigenetic drift theory of aging.
What's that?
So, like,
You mean like what David Sinclair calls the information theory?
yeah, more or less.
All right.
Yeah. So, with time, the gene expression gets more aberrant. So, at what time is incompatible with life.
Okay.
So let's say that we are in a purely damage-based approach, but you don't tackle this epigenetic drift. So don't you think that maybe at what point this gene expression is so awful that it's impossible to correct it with a damage?
This is a very important idea, and it has its roots a long, long time ago, just like pretty much everything in aging. It was originally called the disdifferentiation theory of aging. Richard Cutler, very, very wonderful and perhaps undervalued, one of the grandfathers of gerontology, realized that you would have this epigenetic drift. And he looked for the expression of hemoglobin in the brain, and he found it in neurons, right, in older people, precisely because the gene for hemoglobin was being, inadequately turned off by virtue of loss of methylation or whatever, right? So yes, absolutely. We now absolutely appreciate that epigenetic noise, I prefer to call it that because drift is a little different, epigenetic noise accumulates over time. But then we have to ask, how easy is it to fix? Some people are quite worried about this. In particular, Peter Fedochev has been saying that he feels that one reason why mice are poor models to use for aging is because they are so terribly bad at not aging that we don't really see epigenetic drift as a major driver, whereas he believes in other organisms we do. And he's got pretty good arguments for that, I believe. However, what he doesn't have good arguments for is the idea that this is incredibly hard to fix. He starts out from the idea, well, this is information, and therefore the only way we can fix it is from some kind of external oracle that will, somehow tell the cell which methylation sites are the way they ought to be and which ones have been randomized. But I say, well, wait, how come partial reprogramming works? Partial reprogramming is a rejuvenation of the epigenome. And as such, for me, it is a demonstration that the information has not been lost, or at least most of it has not been lost. It's just being suboptimally used. The analogy that I have been using for a while now to describe this is with a digital image. If you have a digital image, right, 1,000 by 1,000 pixels or whatever, and you randomize half the pixels, right, then it's going to still be recognizable, but the quality of the image will be visually degraded, right? But now, supposing you want to fix it, one thing you could do would be to get a copy of the image from somewhere else and say, well, okay, these pixels are wrong and fix them. That's the oracle approach. But here's another way you could do it. You could just say, well, okay, let's change each pixel in this degraded image to the average of the pixels immediately around it. Now, this means that you'll be changing the randomized pixels and you'll also be changing the unrandomized ones. But it'll still work, simply because in your average image, nearly all the pixels are pretty close to the average of the ones immediately around them, right? So you'll get virtually all of your image back without any oracle. And I believe that that's fundamentally how partial reprogramming works. Basically that it tells the Yamanaka factors come in and they essentially stemify the epigenome a bit. So you started out, let's do it this way. You started out with a cell that's 100% kidney, right? Or 100% neuron. 100% neuron, let's say that, right? And epigenetic noise happens, and by old age, it's become like 80% neuron and 20% random, right? Then you come in with your Yamanaka factors. And what it does is if you let them go completely, it would turn everything into 100% stem, right? But you only do it partially. So let's say you get it to 50% stem. Okay, the things that would turn into stem come from both pools. So what you've got is 50% stem, 40% neuron, 10% random. With me so far? Okay, good. Now what's going to happen? You let go, and the cell looks at itself and says, fuck, I am in a mess. It says, I'm obviously basically a stem cell because I've got all this stemness. But I'm also obviously supposed to be a neuron because I've got all this neuronness. So what it does is it leaps into action and fixes everything as best it can. And it fixes it in a neuronal direction because that's what it knows it's supposed to be. So you end up pretty quickly.
They are tilted to become...
Yeah, so you end up with all the stemness being turned into neurons. And maybe the randomness is still random, but you've only got 10% now rather than the original 20%. So you've got a more neuron-like neuron than you had before you did the partial reprogramming.
I mean, but however...
You have to finish.
Systemic in vivo partial reprogramming experiments have been kind of a failure. So,
I basically disagree. I think that at this point...
For normal mice, for not privileged mice...
I certainly agree that we don't have anywhere near as definitive experiments with partial reprogramming as we have with rapamycin, for example. But give us a year at this point. This is what I'm saying.
I hope that RMR2...
I don't mean give me a year. I mean give the community a year. There's an awful lot of work being done here, and it's going rapidly in the right direction. I'll be very surprised if we don't have, literally 12 months from now, published results showing serious life extension, like a month or two at least, mean and maximum, starting late in life, with partial reprogramming and nothing else.
Alejandro Ocampo was speaking about that in the conference. He talked about that. He's also not as optimistic about you, like you, but he's very optimistic. This is happening in the next 5 years.
So we have to close down soon. We know you have another engagement. I just wanted to ask you if there's happens to be any billionaires in the audience in Spain or internationally. How much money would speed things up really for your work?
Okay, so I've been saying for 20 years that we need more money in the field, but I'm now saying it in a much more targeted way than I was ever saying it before, because we have now reached this point where we can do these combination rejuvenation experiments, which have never been possible until a couple of years ago, and they are astronomically valuable in terms of the amount of information that we will get and the amount of impact we will have on society from success in these experiments. So, there's nothing better. And the thing is, these experiments are particularly hard to fund in other ways. At Sense Research Foundation, my previous foundation, we worked on really hard, really important stuff, but we had the advantage that we were working really early. We were creating intellectual property. We were creating composition of matter stuff. So that meant that we could eventually spin projects out as startup companies. We did that half a dozen times. That was great. This work does not create significant intellectual property. So it relies on pure philanthropy, people giving money away without expecting to ever get it back. Worse than that, it's not fundable by academic sources either, really, because it needs a different kind of funding structure. These are experiments where, first of all, they're very expensive. They cost a few $1 million each. And secondly, we need most of that money at the beginning, because most of the expenses at the beginning, the rejuvenation treatments we do, buying the mice in the first place, things like that. So this is something that academics like. They like to understand why things work the way they do. That's how you get published in Nature and Cell and Science, and that's how you get your next promotion, and that's how you get your next grant application funded. It's very hard to get academic funding sources to put money into things like this. Of course, there's a huge amount of politics around it as well, because there are very rigid structures for how funding decisions are made that are designed to cover people's asses, right? To make sure that nobody is accused of any kind of bias. And no one will make any exception to that. That applies to the government. It applies to people who pretend to be like the government, like evolution. So it's impossible. So we need pure philanthropy on this.
So you guys know where he is, Longevity Escape Velocity Foundation. You know where Natalie is. We're all friends, and we want to channel any big funds that you might want to make this happen quicker. If you want your loved ones not to die.
If you want everyone not to die.
Yeah,
I mean,
people are very selfish about their parents and their children and themselves.
Right.
But,
And their
so,
pets.
especially the very wealthy people, like, what do I do with all this money?
Human race.
There's a good place to put a lot of money so we can get there faster.
Absolutely.
We all want to.
And nonprofits are also tax deductible usually within the
Yeah,
United States.
I should mention
so
that.
You can
So we're a US
deduct
charity.
it off your
So
taxes.
we are a US charity, so anyone who pays taxes in the US can get a tax break on giving us money. But we also have a European charity. It's based in the UK, and so UK taxpayers can have the same thing. But also because of a wonderful entity called Transnational Giving Europe, people all around Europe, including Spain, can do the same thing. Anyone wants to know the details, obviously just ask us and we can tell you.
Awesome. Thank you so much.
Thank you for
this
having
time.
us.
Yeah, it was great.
Yeah.
Thank you so much. Until next time. We will continue the conversation another time.
Thank you.
Immortality!
Thank you, guys.
Rock and roll.
Thank you.
Thanks, Sergio. Good job.
Besitos.
Muy bueno, Sergio.